Chapter 19, Biology Theory Video 7, Gene Editing. Before 2012, when adding a gene, they could not predict where the gene is going to be added. So it could have been even to the middle of another gene that would have resulted in unpredictable consequences. But a technique called CRISPR and Cas9 was developed in 2012, uh, which made it possible to edit genes. So this gene editing uh, has been used not only in bacterial or other simple cells, but also in animal cells to edit the genetic code. So here in CRISPR, there is a group of base sequences that code for short lengths of RNA that direct a nuclease enzyme known as Cas9 towards specific base sequence. So the CRISPR associated Cas enzyme is an endonuclease that cuts DNA at a point that is determined by these RNA molecules, known as guide RNA, gRNA. Part of each guide RNA has a sequence of 20 bases that can locate and bind to a strand of DNA with a complementary base sequence. The enzyme Cas9 has two active sites to cut DNA across both strands. So here, all of these, this is a explanation of a very complex process. You do not need to know the process, but you need to know how this CRISPR and Cas9 system can be used for our benefit and to correct any changes that need to be done in DNA. So the CRISPR-Cas9 system is developed so that gRNA, that is a guide RNA, can be made with the base sequence that is going to be complementary to any base sequence of DNA. So that gives the genetic engineer a way to edit the DNA at specific places anywhere within the organism's genome. So when it is cut by Cas9, the natural DNA repair mechanism can repair the break. This can be done by adding one or more nucleotides that change the base sequence. That means it can add in a short length of a prepared double-stranded DNA. That means you can add a G into that place, but the difference is you know where you are going to add this to. So it is going to be a known position. So it will here, as you can see here, DNA is repaired by mechanism that inserts a base sequence to silence the allele, or it inserts a short length of DNA to correct faulty allele. So that is the process. So it can be used to remove DNA, that is to remove a faulty allele, or to removing part of the faulty allele. So that means instead of the wrong or the misfunctioning protein, it would not code for anything that functions. So that means that faulty property would not be expressed. Then also this gene editing can also be used to replace a faulty allele with a functioning one, or replacing part of a faulty allele so that it then codes for the functioning protein. So you can use the toolkit that cut and paste genes or parts of genes with a better precision than what they did using the vectors. Write down a note on this. gene editing. This is a form of genetic engineering in which the genome of an organism can be changed by deleting, inserting, or replacing a length of DNA by using a method such as the CRISPR-Cas9 system. In CRISPR-Cas9 system, a group of base sequences that code for short lengths of RNA that direct a nuclease enzyme known as Cas9 towards specific base sequences. And they can cut DNA at a point determined by these RNA molecules known as guide RNA, gRNA. This CRISPR-Cas9 system is developed so that gRNA can be made with base sequences that are complementary to any base sequence of DNA. As a result, genetic engineer will be able to edit DNA at specific places anywhere within the organism genome. After Cas9 has cut the DNA, natural DNA repair mechanism will repair the break by number 1, adding one or more nucleotides that change the base sequence. Number 2, inserting a short length of prepared double-stranded DNA with a specific base sequence. I would suggest that you pause the video and draw this figure 19.10 so that you would understand it better. CRISPR-Cas9 can be used to remove DNA which are faulty or would remove part of the faulty allele so that it would not code for any malfunctioning protein, or else edit the genome so that it would produce the proper functioning genome. CRISPR-Cas9 is actually short for clustered regularly interspaced short palindromic repeats plus CRISPR associated protein 9. So these base sequences are said to be found naturally within the genomes of prokaryotic organisms. That means both the bacteria and archaea. So these sequences come from DNA fragments in the phages that infect the prokaryotes. So they can be used to detect and destroy DNA from similar phages that attack the prokaryotes. So the process is by producing gRNA, that is a guide RNA that would direct the nucleose enzyme Cas9 to break so this is just like the cutting mechanism that we discussed earlier. Cas9 to break sugar phosphate backbone of phage DNA. So it will prevent the virus from taking over the phage DNA. It will stop the virus from taking over the prokaryote. so this so as a result, we can suggest that the CRISPR-Cas9 enzyme will have similar function to CRISPR-Cas9 will have a similar function to the enzymes that we discussed earlier, the restriction enzymes. So it provides the protection against viruses. Then also these Cas proteins have the ability of integrating the parts of the viral DNA by inserting them into the CRISPR sequence. So that means it will have a memory of the past infections because they have the base sequence that are derived from the infection. So when the bacteria is reproducing, it will actually get passed on to the next generation. So in 2012, Dr. Emmanuel Carpentier and Dr. Jennifer Doudna, they used this CRISPR-Cas9 as a tool to edit genomes. They have shown that the guide RNA, if the guide RNA is changed, then Cas9 will cut DNA at a site that is going to be complementary to the gRNA sequence. So that means it can be made a custom made endonuclease. That means we can make it the way we want. So in the restriction endonucleases, there were restrictions because there were limitations that we could not predict what was going to happen. So as a result, we can actually the possibilities of this is going to be medical users and the pre-medical users. That means before a disease comes, we can change the genome and we can entirely get rid of it. We can this is also there is also a possibility of making customized drugs to treat cancers. Then we can change the genomes of insect vectors to prevent them from spreading diseases such as malaria. Then we can make sure that the bacteria that are used in the industries will be protected against viruses. Then they can also be used to make sure that human embryos do not have defects. So you can delete certain parts of the gene code and destroy them, or we can destroy them in order to prevent prevent some of the diseases. Now, for example, this in China, Chinese some researchers had declared that they have used this CRISPR-Cas9 system to delete part of the gene coding for cell surface receptors known as CCR5. So HIV would gain entry to cells through this by binding to this. So what they have done is, so when they deleted this code, they have made the children resistant to HIV. So this is going to be a little bit of an ethical problem also, because you can change when you can change the DNA sequences, people will in the future will try to make people who are similar to Superman or Superwoman, because everything would be perfect. They would make everyone without any defect. So that could be a result, and possibly studies are being also done in that regard. Write down CRISPR-Cas9 system is developed in order to change the genetic code or the genome of a cell by changing the guide RNA by Cas9, which will cut DNA at a site complementary to the gRNA sequence. As a result, we are able to sequence the genome of individual people and many possibilities come into play. Right. I hope you understood. This is a bit of a complex area. Please read through that again, and if you do not understand, please let me know.